So this is going to be our first lecture about bacterial pathogens, and we're starting with gram-positive. More specifically, we're going to start with gram-positive cocci. So under the microscope, these are going to be purple circles. And the two organisms that we're going to be focusing on first are the Staphylococcus group and the Streptococcus group. So that's what we're going to do. As this shows, there's a variety of gram-positive pathogens. So remember that that stains purple. And they can be broken down into two larger groups. The group that we're going to be focusing on first, as I mentioned, is this staph and the strep group. There are other gram-positive, and these names hopefully are familiar to you. We've talked about Bacillus and Clostridium prior because those are endospore formers. And we've also talked about Mycobacterium because those are the acid-fast bacteria that include tuberculosis and leprosy. So these are broken down based on how at least one component of their DNA or their genome. And so you can measure how much, remember the base pairs. So A pairs with T and G pairs with C. So you can look at the relative abundance of GC pairs in a given genome. And so there's a group of gram-positive bacteria that are classified as low GC content. So that means they have more A's and T's than they have G's and C's. But then there's also ones that are classified as high. So we're going to focus on this, again, the staph and the strep group, starting with the Staphylococcus. So we've talked about Staphylococcus before because it's an arrangement. And so these guys are gram-positive cocci, so purple circles. And they appear in grape-like clusters. And remember why we get arrangements. So when some bacteria divide, they fail to completely separate, and so you get cells stuck to one another. And because Staphylococcus divides in kind of an irregular way in terms of cleavage planes, you can get this, what we call irregular clusters or grape-like clusters of them. So this picture down here is just reminding us of how they can be stuck together and they look like a cluster of grapes. There's some other characteristics that we've touched upon at different times. So salt tolerant describes the Staphylococcus group. And that characteristic is one of the components that allows them to colonize and survive on human skin. And so if you think back to a lab exercise that we did, we used a media that was called MSA for short, and that's mannitol salt agar. And so that second word in there indicates the high salt concentration. And so if you, when you swabbed your skin or your nasal passage, and if you got growth, then that growth is likely Staphylococcus growing on your skin or in your nose. And then, as we'll see in a little bit, there is another part of that media, the mannitol portion, that allowed us to say if it was which of these two species it is. And so there are two staphylococcal species that we're interested in. So Staphylococcus aureus is one, and then Staphylococcus epidermidis is another. Both of these can be found, as I mentioned, on human skin, or so they're part of the microbiome. And so that makes them what we classify as opportunistic pathogens. They're going to be common wound pathogens that can cause infections of wounds that we'll see. But skin infections are one of the common ways that a staph infection can kind of take hold. One last thing that we have on here, we've seen this term before when we were talking about infectious disease. So fomite is again that just non-living object that microbes can survive on. And so Staphylococcus is one in which they are pretty resistant to drying out, or desiccation is another word for that. So that allows them, so if they are on somebody's skin, they can survive off the host for some period of time. And this also, as we'll see later on, allows them to be an important cause of nosocomial infections, those healthcare acquired infections that we've talked about before as well. So the reason why Staphylococcus can cause disease is because of topics that we talked about before. I'm not sure why I have 3 here because I have a list of 4. Sorry about that. And these are known as virulence factors. So remember the categories of virulence factors that we saw in a previous lecture entitled infectious disease. And so if these microorganisms, these Staphylococcus, can breach that outer barrier, that skin, then entry of not that many microorganisms, so a few hundred cells, can progress to a disease state. And that's because of these different virulence factors that exist. And so these reflect the categories we saw before. So adhesion factors is one, anti-phagocytic factors is another category, enzymes and toxins were all examples of different types of virulence factors. So most of these virulence factors that Staphylococcus has, we have subsequent slides that we're going to talk about. But I do just want to point out this one now because this is the only place that it's talked about. So one of the important adhesion factors that was introduced was this term. It's called adhesin. And if you don't remember, I mean, you can go look back in that PowerPoint, but I'll just put it on here. These are going to be proteins on surface of bacteria for attachment. So that's what adhesins are functioning to do. And so Staphylococcus has these proteins on the surface for attachment called adhesins. Before we get into this list of virulence factors, I do want to introduce this concept to you. So there are different strains of Staphylococcus, and again, mostly what we're talking about here when we talk about a staph infection, it's most likely going to be this one, Staphylococcus aureus. So there is not all Staph aureus are exactly the same, and so the way that the term we use to describe the variations that exist is we say, oh, this is a specific strain of Staph aureus. So there's commonalities between all Staph aureus, that's why it's called Staph aureus, but they're not all identical. And so the word strain is used to capture those specific differences of pretty similar microorganisms. And so because there are so many virulence factors that exist across all the Staphylococcus aureus strains, what's going to determine the clinical manifestation of a staph disease is going to be very much dependent upon what virulence factors are present in a particular strain of Staph aureus. And that also makes staph a little bit more difficult to diagnose because there's not one disease that it causes, so it's not a nice one-to-one relationship where we can say something like, I'm trying to think of an example, but there's one bacteria called Clostridium botulinum that causes botulism. There's different types of botulism, but for the most part, it's pretty similar across the board. Or Bacillus anthracis causes anthrax, etc. And so there's these specific diseases that some bacteria cause, and pertussis causes whooping cough, etc. Staphylococcus is a little bit more tricky, but it also is what makes it really important because it causes so many different diseases, and it's a common human pathogen that we want to be able to know how to identify it across these different clinical manifestations. So that's what we're going to do over the next several slides. We're going to learn the different possible virulence factors that strains can have, and they're going to be in these remaining categories that we haven't yet talked about in much detail. And then we'll move on to the types of diseases that this particular organism can indeed cause. So we're going to start with anti-phagocytic factors, and so that's what we have here. And so there are 4 different anti-phagocytic factors that can be present in different strains. So the first one that's commonly found across most strains of Staph aureus is a protein on the surface that's called protein A. So this is a really important virulence factor in Staph aureus. And it has several functions, but its primary function is going to be to, again, disrupt how the host immune system responds. So we haven't talked about them in great detail, but hopefully you know from other classes or just know because it's a pretty important way by which our immune system functions is we have the ability to generate these molecules called antibodies. And antibodies are supposed to bind specific microbes. And then in doing so, there's a different mechanisms, but ultimately those destroy those microbes after antibodies bind them up, they're going to be destroyed. So antibodies are a really important part of how our immune system functions, and so interfering with how those antibodies bind is going to then diminish how well our immune system is functioning. And then also, phagocytosis is another really important mechanism that our immune system uses, and some of this phagocytosis is related to antibody function, so these kind of go hand in hand. But antibodies and phagocytosis are two really important mechanisms of how how the host immune system is going to be functioning within a human. And so this protein on the surface causes disruptions in several important host immune cell mechanisms. So that's one really important anti-phagocytic defense that Staph, and this is mostly Staph aureus. So in parentheses, I have which of the two Staph species frequently exhibits these specific virulence factors. Now, this coagulase is an enzyme, and so we're going to see this on another slide as well. But this enzyme functions in a specific way, as we learned in a previous lecture, that infectious disease lecture. And so Staph aureus has this enzyme, coagulase, that allows it to form blood clots around itself and basically hide inside that clot. And the reason that's important is, again, immune cells are going around trying to ingest and digest. That's that process of phagocytosis. So for that to be functional, the white blood cells have to find and then ingest and then digest the bacteria. And so what we're saying here is if the bacteria are hiding inside of a blood clot, then those white blood cells don't have access and won't be able to find and then destroy the staph organism. Capsules are one of the two types of glycocalyces that we talked about. So it's a glycocalyx. And we said when we learned about them, capsules cause the host immune system to not be able to function as properly. So phagocytic cells can't function as well. And so these capsules on Staphylococcus are going to, again, inhibit phagocytosis by white blood cells. And then there's some stickiness that occurs with capsules. We primarily think of capsules as being important for hiding from the immune system. But capsules, because they're this outer polysaccharide and protein mixture, they can have some stickiness to them. So these capsules also allow Staphylococcus to attach to and colonize artificial surfaces. So that also explains how they can be important nosocomial infections, because they can get onto catheters and things like that where they can cause infection. And then the last, we've seen this before, leuco relates to leukocytes or white blood cells. And then we see this ending side or cidal. So leukocidins are going to be chemicals that can kill white blood cells. And so white blood cells, again, are the important phagocytic cells. So some Staphylococcus can and does make these chemicals that can destroy white blood cells. So all of these have the capacity to disrupt host immune response in some way. So then the question becomes, how many of these does a particular strain have? And obviously the more of these that they have, the harder it is that the immune system is going to be able to destroy that. So now we're moving on to enzymes, and the first one we kind of already talked about. So I just have it on this one just as a reminder, as it's an enzyme. But now we have these other enzymes. All of them end in ase, as we've seen. And so hyaluronidase we've talked about, again, in the infectious disease. So many Staph aureus strains have hyaluronidase. And if you don't remember what that is, hyaluronic acid is one I call it the glue that's between our cells. So it has other names like extracellular matrix, but hyaluronic acid is important component of that glue, of that connective tissue that's holding our cells and tissues together. And so if bacteria make hyaluronidase that breaks down this glue, it enables bacteria to invade beyond specific sites where it might start. So again, if this starts as a skin infection, but the strain has a hyaluronidase, then it may be able to actually infiltrate and invade beyond just where that wound is, and then eventually get in the bloodstream, and then disseminate and spread throughout the body, leading to other more serious and systemic infections. Kinase is an enzyme that's frequently paired with coagulase, but kinase in this case is called staphylokinase because it's in Staphylococcus. And so these kinases dissolve the clots, and so this allows Staph aureus to free itself from clots, whether it's clots that they made or clots that our body is making at a wound site to try to keep things kind of walled off there. So staph can, again, many of these start as wound infections, so they're on your skin and you get a cut, and the bacteria are now getting deeper into the epidermis and the dermis. And then part of that damage, we form clots, and that helps not only stop the bleeding, but also kind of wall off that area and try to prevent things from getting into the bloodstream. But if bacteria has a kinase, it can dissolve that clot and try to invade further. So we have several of these enzymes now that allow the bacteria potentially to invade, which makes them obviously better able to cause disease and spread. We have this enzyme that we've seen before. It was way back when we were talking about antibiotics, because there is a certain category of antibiotics that includes penicillin that are called beta-lactams. And so that's a whole category of antibiotics. Penicillin was kind of the first and probably the most often used of the beta-lactams. But Staphylococcus aureus, most strains anyways, 90-something% of them have this enzyme called beta-lactamase that we know will destroy or break down penicillin. So not surprisingly, if someone has a staph infection, penicillin is not a good choice to treat, because most staph infections caused by Staph aureus are going to have this enzyme that just destroys penicillin. And so we'll talk about how to treat staph infections, but we can talk about it at one level now and say, well, don't use penicillin to treat staph infections. And these are just kind of a collection. Hopefully you see at the beginning of this lipases. In this, it looks like lipids, proteases, looks like proteins, and deoxyribonucleases, that looks like DNA. That's protein. That's lipids. So these are all enzymes that digest or destroy each of those things. And so many staph strains have lipases, deoxyribonucleases, and proteases, and that enables them to digest those different components where they can get nutrients from the host, since all cells, including humans, are going to have lipids, proteins, and nucleic acids that are present. So it helps explain how bacteria can survive and thrive not only on our skin, but then also as they invade other tissues potentially during a course of an infection. The last virulence factor category is toxins. And there's a variety of toxins that, again, not every strain of staph makes, but some do. And so depending upon how many of these toxins are produced, will explain how the clinical manifestation presents itself. So what I wanted to do first is remind us what we're talking about. Whoops. So when we were introduced to toxins in the infectious disease, there are two major categories. There are exotoxins and there are endotoxins. So all of these toxins that we're talking about here These are all exotoxins, and if you don't remember what that means, it means that living bacteria make and release. So that's what we're talking about here. So there are some strains of Staphylococcus that can make and release these toxins called enterotoxins, and other ones can make cellulitic, and others can make toxic shock syndrome toxin, or TSST, or exfoliative toxins, etc. The endotoxin only in gram-negative bacteria. So we're not going to have to think about the clinical manifestation of the effects of endotoxins until we get to gram-negative infections. But since we're talking about gram-positive cocci, that's the group we're talking about, any toxin by definition is going to have to be exotoxins. So let's go through these different toxins. So enterotoxin is a subtype of exotoxins, and it's describing where these toxins are going to affect the patient. So enterotoxins are going to affect, they target the GI tract. And so there are some strains of Staphylococcus that can cause food poisoning, which can be described as there. So you can have intense contractions and pain of the stomach and the intestine, and then nausea and vomiting. And this is because of these enterotoxins that are produced. I have this here. It says heat stable. And so 100 degrees Celsius is boiling. So that's what water boils at. And so 30 minutes of boiling is a pretty long cook time. And so the bacteria may actually be killed. But if this toxin has been generated prior to cooking, then even though you kill the bacteria, you still might have enough enterotoxins that can make you sick. And so this is one of the reasons why it's important in terms of how long you have food sitting out on counters prior to either cooking or after cooking, and how we thaw different meats and things like that, that it's important not to just have things sitting at room temperatures for extended period of time before consumption or prior to cooking. And that's because of these staph enterotoxins that can cause food poisoning. Even though you may still cook the food and destroy the bacteria, the toxin may be present. Cytolytic toxins, that just means toxins that are going to cause cell lysis. So cyto means cell, and lytic means lysis, or to break open. And so there are some toxins that are going to do just that, and they do so by targeting that cell membrane of host cells and tissues. So cytolytic toxins are just going to be kind of generic in terms of they just lyse cells. These are specific examples that some strains have. So we've talked about leukocidins, the cell membranes of white blood cells, or leukocytes, is what they're going to target. But then we also have something called hemolysins. We'll talk more about these in the strep category. But hemo relates to blood, and then there's that word lyse again. So hemolysins are going to lyse red blood cells. Well, they're going to lyse blood cells. Most of those blood cells are red. They can also do white blood cells too. So they have some leukocidin capacity to them. The next couple of toxins cause very specific clinical manifestations. So some strains produce toxic shock syndrome toxin, and that will cause toxic shock. And the signs and symptoms are listed over here. So there's a lot of things that are going on with toxic shock syndrome, and it's related to the production of this toxin that is what we call a super antigen, and all that means is our immune system is going to be over stimulated. And so this one toxin is going to cause our immune system to basically go into overload and overdrive, and it produces so many different immune signaling chemicals that it negatively disrupts a lot of our physiology. And so then that's manifested in a lot of these different signs and symptoms of disease. So staph is one of the primary causes of toxic shock syndrome because of the presence of this toxin, but there's also a streptococcal variety as well. So those are the two primary organisms that can cause toxic shock syndrome. And the last toxin that we'll talk about is exfoliative toxin. And you've seen that word before. It's kind of like to remove skin cells by, you can use like an exfoliative scrub or something like that. But this obviously goes beyond that. This is a toxin that's going to damage the anchors that holds your upper layers of your skin kind of intact. And so when this toxin operates, it's going to cause separation and sloughing off of the skin. And so this is an example of an infant that has a staph infection in which the exfoliative toxin is present. And so it looks like a really bad sunburn, or the after effects of a really bad sunburn where you have sheets of skin that are peeling off. And this is because of this exfoliative toxin.
So based on what we just talked about, there's really three major categories of staphylococcal disease. So the first is that food poisoning we were talking about. This is known as non-invasive because the bacteria don't actually have to enter your body. You're only ingesting that specific toxin, so that enterotoxin contaminated food. So again, even if the food is cooked thoroughly, if during preparation there wasn't sufficient caution taken, that enterotoxin can accumulate. And then even after cooking, you can be ingesting that. And so the bacteria don't need to invade. You just have to eat that enterotoxin contaminated food. This is how we most often see many staph infections, is some cutaneous manifestation. And there's so many different types of clinical manifestations of this. So we saw one of these. So that baby with the peeling skin is called scalded skin syndrome. And that's a result of that exfoliative toxin. Impetigo is kind of, a skin lesion frequently seen in children that's kind of pus and crusty. And then there's folliculitis and these other skin diseases as well. And so there are a variety of photos you can look up. And I mentioned the surgical wound sites, or just other wound sites in general, we know can be a location where staph can kind of start an infection and develop to a disease state. And the last and kind of most serious is a systemic disease. And that just means that the bacteria or the toxins have gotten beyond a certain local point of infection and gotten into the bloodstream. So again, either the bacteria or the toxin have gotten into the bloodstream. And so the word we use to describe bacteria in the blood is that bacteremia. And once that occurs, again, bacteria can end up in a variety of places, whether that is the lungs or in the bone. Also, we can have some issues with the heart. And then if the toxin is traveling through the bloodstream, we have at least that one specific toxin, the toxic shock syndrome toxin, that can lead to toxic shock syndrome. That's a systemic disease that affects a variety of physiological systems, as we saw on a previous slide. So in terms of diagnosing, treating, and preventing, there's because there's so many different types of clinical manifestations, how are you going to know if staph is the cause of this? And another way of looking at it also is, for each of the diseases that are presented, staph isn't the only causative agent of it. And so it could be staph, but it could be, say, for example, strep. So how can you say definitively that, oh, this pneumonia is caused by staph, or this skin infection is caused by staph? So one answer to that, this is very similar to what we saw for the fungal infections. So a microscopic analysis. So as we said, Staphylococcus is going to have a very characteristic look under the microscope. So gram-positive, I guess I'm missing something here, cocci in grape-like clusters. So that's pretty important. So that can be isolated from different regions of the patient, depending on how it's clinically manifesting itself, but then also culture-based approaches as well. And so we saw the plate that we used, the mannitol salt agar. Staphylococcus aureus is one of the few that will grow in the presence of high salt, and it ferments mannitol. And so getting that result is pretty indicative that's Staph aureus. We also saw that Staph, we'll see one really important clinical differentiation between Staph and Strep is their response to catalase. So all Staphylococcus are going to be catalase positive, and one important designation, all Strep are going to be catalase negative. And so that's a really important distinction between Staph and Strep infections when it comes to culturing and then doing biochemical tests. And then also coagulase positive is another important characteristic of Staphylococcus, especially Staph aureus anyways. So these three results through culture-based and biochemical tests are really critical in identifying something as Staphylococcus. And then also there's that microscopic evaluation as well. As we mentioned earlier, this enzyme that many Staph aureus strains have, beta-lactamase, is going to inactivate penicillin or destroy penicillin. And so for that reason, penicillin is not going to be used for staph infections. And so we use some semi-synthetic form of penicillin, so whether it's methicillin or amoxicillin or something like that. But many of you are aware of this acronym, methicillin-resistant Staphylococcus aureus, or MRSA. So that is a Staph aureus strain that's resistant to not only penicillin, but all of these other derivatives as well. And so that means there's less options for treatment. And so one option that we have left is a completely different cell wall synthesis inhibiting drug called vancomycin. This isn't on the slides, obviously, but we now have a growing concern of this strain, which is vancomycin-resistant Staph aureus. And so that means it's a strain that's resistant to penicillin and methicillin and all derivatives and vancomycin. So this one is really problematic, and we don't have much in our arsenal of antibiotics to treat that once it's kind of evolved to that state. So there's not a lot of these strains, but we're starting to see them increase. So in terms of prevention, because staph can be found on people's skin, it's really critical that hand washing and disinfection is part of the procedures, and so preventing those healthcare acquired infections. And then also, because of the virulence factors that we've talked about, the ability to attach to things and colonize and survive on inanimate objects, those things called fomates, it's really important to clean and sanitize things well prior to their use. So that brings us to Streptococcus. And Streptococcus is another gram-positive cocci, but this is going to be organized not in clusters, but most often in chains, but it also can be in pairs. So unfortunately, Diplococcus is what we call the arrangement in pairs, so it's just two circles together. So that's how some Streptococcus species are. The arrangement called Streptococcus is what we have in chains. So a good number of Streptococcus are indeed in the arrangement of Streptococcus, but just realize that some are in the Diplococcus arrangement. And here's a continuation of what I said earlier. Streptococcus, in addition to having that look under the microscope, one important characteristic, biochemical characteristic that differentiates them from the staph that we just talked about, is being catalase negative. So that's a really critical biochemical test that allows to differentiate. There's one other kind of biochemical test that's really critical to identify Streptococcus as well, because there's a series of unique molecules on the surface. These are called Lancefield antigens, and this is after the scientist who discovered them. So Lancefield antigens, they're identified by a letter. So it's Lancefield antigen A and B and C and etc. So there's a variety of different Lancefield groups. or they're just called Group A Streptococcus or Group B Streptococcus or Group C, etc. There's only 2 that we know can cause disease within humans to any significant extent. And by far the most important is Group A Streptococcus. And so it often is just abbreviated as this, Group A Streptococcus. And although it's a group, it's called Group A, there's really only one member of the group. And so this particular genus and species, so Streptococcus pyogenes, is the one Group A strep organism that we're going to talk about. And it's the causative agent of a variety of diseases, but most commonly we see it as the causative agent of strep throat. So Group A Streptococcus is what can cause and what does cause strep throat, but there's a lot more diseases as well. Group B is Streptococcus agalactiae, but it's not as common, so we aren't necessarily going to spend time talking about that one. So Group A Streptococcus, so it has that A Lancefield antigen, and so that's something that we can test for as well. So when we're going to talk about diagnosis, we'll see that. But the reason it's called Group A strep is because it has this unique molecule on the surface called the A antigen. It also has a certain look under the microscope, as we said. So they're going to be cocci in chains. So this is what they look like. And then when they grow, they have a certain characteristic as well. So when we incubate and culture them, so they're going to have white colonies, and they're going to exhibit this pattern on what's called a blood auger. Since we're kind of remote this semester, you guys haven't used blood auger plates, but as you might guess, it's going to be auger that has blood infused into it, so that's why it's got this red color. And different bacteria have the abilities to break down blood to different extents. And so when we call something beta hemolytic, or it exhibits beta hemolysis, if you look at that word, hemolysis, that's blood, lysis or breakdown of blood. And so beta hemolysis is the complete hemolysis of blood. And so this that I'm kind of tracing right now, that's the bacteria that was put onto this plate. And what's clearly surrounding it is this area where the blood color is completely destroyed, because this particular bacteria has the ability to completely hemolyze the blood. And so we classify that bacteria as beta hemolytic. And that's what strep pyogenes looks like on a blood auger plate. So that's a really important characteristic of them. Just to contrast, not all bacteria can do this. Some are classified as what are called alpha hemolysis. So alpha hemolysis means the bacteria partially break down the blood or partially hemolyze the blood. And what you get is this kind of green discoloration. So if you look right here, there's no bacteria, but you can kind of see the red, original red color of blood is changing to this kind of greenish tint. So if you kind of look here, that's the original red color, but then as you get close to the bacteria, you can kind of see this green tint. And that's because this bacteria would be classified as alpha hemolytic. And then bacteria that are classified as gamma hemolytic, it means they can't do anything to the blood. So they grow on the blood, but there's no effect to that red pigment at all. So this pattern of hemolysis is something we use to describe bacteria. So it's another kind of box or category that we can put them into, but strep pyogenes are going to be, as we mentioned, beta hemolytic, which has an important meaning. We also have a variety of different strains of Group A Streptococcus, or strains of strep pyogenes. And so that's going to influence, again, the diversity of signs and symptoms and the diversity of diseases that we can get. But one thing that we can say is that most of the pathogenic strains, so the ones that can cause disease, are going to have a capsule. So remember what capsules allow bacteria to do, which is to avoid the immune system. And Group A Streptococcus or strep pyogenes, like Staph aureus, is a really important human pathogen. That's why we're spending some time talking about them. So this is one of the more common pathogens that we see. We can do a similar analysis of Group A Streptococcus virulence factors. and I'm going to jump to the bottom first. So there's a bunch of different strains for group A, and they each have different virulence factor combinations, and so that is what allows for group A to cause such a diversity of diseases. So we have the same kind of categories: adhesion factors, anti-phagocytic factors, enzymes, and toxins. And there's some specific examples of each for, in this case, it's Strep pyogenes. So it does have adhesins. There's a specific protein that we're going to see twice that's really important. It's called M protein, sometimes called protein M. But this one protein on the surface of group A Streptococcus has two really important functions. So it's going to be an adhesin for attachment, but it also is going to be, since it's on the surface, it can deter that process of phagocytosis. So it really has two functions in one, which makes it a really important virulence factor. We mentioned most pathogenic strains have a capsule. This particular bacteria, Strep pyogenes, or group A strep, has a unique capsule in that one of its principal components is hyaluronic acid. And that should sound familiar to you because we've talked about hyaluronic acid before. It is that glue between cells. But and I guess what I should say then is it's a normal molecule within the human body. And so this bacteria has a capsule made out of a molecule that is found throughout the human body. And so this really masks the bacteria, making it harder for our immune system to recognize these bacterial cells as being foreign because it has this really common molecule that we have throughout our body on it. So it helps to camouflage the bacteria. We've talked about leukocytins. So again, this breaks down white blood cells. These actually have what are called streptolysins. And so streptolysins is what explains that beta hemolysis that we just saw. So we said beta hemolysis is the complete breakdown of blood, and so streptolysins can and do break down red blood cells, but they also break down and destroy white blood cells as well. And white blood cells, as we have talked about multiple times now, are the immune cells, and they are the important phagocytic cells. And so by destroying those, that's going to be it could play an important role in serving as anti-phagocytic factors. So this is the only one that's kind of new we haven't talked about, but you should be able to notice that is an enzyme. And so this is a bacterial enzyme that breaks down a member of our complement. And so we haven't talked about complement proteins yet, but complement is a series of proteins that helps our immune system function in different ways. And so C5a peptidase is a bacterial enzyme that breaks down an important complement. This particular complement's role is to help allow phagocytic cells to move to an infection site. So it's basically, you can think of it as like an alarm call, like a call to 911 and come to this address, etc. in our body. And this bacterial enzyme is going to destroy that signal. So the immune response is not as coordinated, and so it's harder then for the immune system to respond appropriately. So those are anti-phagocytic factors. We've seen all of these enzymes before, and so I'll just do a really quick review. Hyaluronidase breaks down hyaluronic acid, which is the glue between cells, so it's important for invasion. Kinases break down clots, so this isn't called staphylokinase, it's called streptokinase. So this, again, if there is a wound that a clot is forming that's trying to wall off that infection site, there are some bacteria, so strep, group A strep included, have streptokinases that can break down that clot, which leads to invasion. And then we've seen these proteins like we saw in staph. This says deoxynucleases. I think I had deoxyribonucleases before, but they're the same thing. They have several different names. Unfortunately, I didn't catch that until I was just reading it out loud. So deoxynucleases or deoxyribonucleases and proteases This is going to break down DNA. This is going to break down proteins, and it allows these bacteria to use those molecules as nutrients, and also to destroy structures that exist within the cell that allow invasiveness of the bacteria. The last category here is toxins. And these are called pyrogenic toxins. So notice we've seen that prefix before. It relates to fire. So this is fire generating. But it really is talking about fever generating. So that's what we mean by pyrogenic. So these toxins are also going to serve as superantigens. Let me back up for a second and just remind you again. So these, all the toxins we're talking about here are exotoxins. So that means made by, released from living cells. And these pyrogenic toxins serve as superantigens, and what that means is overstimulates the immune system. And so it kind of causes the immune system to overreact, and you can get an overabundance of response. So you can get a pretty extreme fever. You can get other things like toxic shock syndrome, again, depending upon what pyrogenic toxin it is. But it's caused by these toxins overstimulating the immune system and the release of these immune modulating chemicals called cytokines. And so the cytokines are released just excessively, leading to all sorts of physiological changes that are part of the disease. So there's a bunch of diseases. Sorry, I'm going to pull these up. So strep throat is probably one of the more common manifestations of this, and so you can have redness and swelling of the throat. This same bacteria, we know it's associated with other infections that it can progress to. So one is called scarlet fever. We don't see this as much now because of antibiotics, but this was a common progression of this disease. We're actually seeing an uptick of it now because of antibiotic resistant streptococcus, so we can't get rid of the strep throat as rapidly, and so you get this rash that occurs over almost the entire body, and this is caused by that pyrogenic toxin that gets in the blood, and it leads to this redness and inflammation occurring throughout the body. Group A is also known to cause ear infections and sinus infections, so that's can be related to strep throat, or it can be on its own. Impetigo we saw under Staphylococcus, which is this crusty scab, pus, lesions often on the face, but can be other areas as well. And then toxic shock syndrome. This is one thing that we can see is the formation of a rash across the body. And so they've outlined it here in pen, and they're seeing if it's spreading or not. And this same sort of thing, it's a toxic shock syndrome toxin, so it's one of those super antigens, pyrogenic toxins from group A streptococcus. So if the bacteria or the toxin gets into the bloodstream, you can get toxic shock syndrome caused by group A streptococcus. There's other diseases as well. So in the news, it's called flesh-eating streptococcus, but the more medical term that we use is necrotizing fasciitis. And so this is, again, one of those, I should change this, I didn't catch this. So same thing, it's one of those pyrogenic toxins that can, along with some enzymes, destroy deep connective tissues, so including muscles and fat. So it's not just a superficial wound, but it's actually kind of destroying tissue deep into the fascia, which is connective tissue. So obviously this picture of a hand doesn't look pretty, and so that is a result of a group A strep infection that has certain toxins and enzymes that allow it to kind of destroy that tissue. And these last two are a little bit unique. Well, I guess I should comment on this. This is after some treatment where they've had to, surgically debride some of that tissue and do some skin grafting. So it's getting better, but there can be, significant damage that may require amputation depending on the severity of the strep infection. These next two are both inflammatory responses. So these are when your own immune system is responding to group A strep disease, there's some molecules that are similar in our body. So after your immune system has responded to group A strep, it may start misidentifying some parts of your heart as being foreign, and you can develop something called rheumatic fever. Or you may also have some obstruction to flow in your kidneys. So this is glomerulonephritis. So if you've taken anatomy and physiology, we know the nephron is the unit of filtration in the kidney, and the glomerulus is where the filtration actually is occurring. And so you can get inflammation and blocking of that, which can lead to some kidney issues as well. So like we saw for Staphylococcus, group A strep can cause a diversity of diseases because there's so many different strains and there's a bunch of different virulence factors that exist. And so it really is a question of which group A strep you're exposed to and what virulence factors exist in it. So what I'm not saying is that group A strep that causes strep throat is the exact same thing that causes necrotizing fasciitis. It's not. They're different strains. But if you get a cut that is has some of this particular strain of group A strep that has the ability to make this specific toxin and specific enzymes that can digest that deep connective tissue, then you could get this sort of, as it's called in the press, flesh-eating streptococcus. So as we saw for Staphylococcus, how do we correctly diagnose this? So you can use similar techniques where we can use a microscopic approach. And so these are, again, gram-positive cocci, but they're going to be in short chains or pairs. In terms of culture-based methods, we talked about on a blood agar plate, seeing that beta hemolytic pattern is pretty important. And then the biochemical test to contrast it with Staphylococcus is catalase negative. But probably the most important way is right here. And that's doing what's called the Lancefield, it's called Lancefield agglutination test, or just a Lancefield grouping test. So remember what group A strep means. It means has the A antigen. And so basically these tests, these are called immunological tests. These tests are going to test for the presence of that A antigen. So as this says, just saying that streptococcus, so if they look streptococcus in your throat, that doesn't mean you have strep throat, because we have streptococcus as part of the normal microbiota that exists there. And so what you're looking for is, are there any strep that have the A antigen present? Because those aren't normally part of the microbiota. So this rapid strep test is what they're doing oftentimes with those throat swabs. Penicillin works for most strains, so that's different from Staphylococcus as well. And then prevention, you probably experienced this. It is possible to get strep throat multiple times throughout your life, so you do generate protection, but there's a bunch of different M proteins that are out there, which is one of the important targets of our immune response. And so because of the diversity of strains, you don't have protection to all strep from one exposure. And so over time, and with each exposure to disease, you can get some protection that then allows you to be protected from an increasing number of strains as you expose to different ones over your life.